Okay, working out the genetic code. But after the discovery of Watson and Crick of the double helix, and with progress in understanding that amino acids made up proteins, and proteins had a very specific sequence of amino acids, it became clear that probably the sequence of bases in DNA somehow coded for the sequence of amino acids in proteins. But of course, we've only got 4 bases in DNA, but there are 20 different naturally occurring amino acids. So if you think, then it became clear that obviously it wasn't 1 base per amino acid. It couldn't even be 2 bases per amino acid, because that would give you a maximum of 16 4 squared possibilities. It's not enough, not quite enough. But if you have 3 bases and 3 bases code for 1 amino acid, then that would give you 64, which would be enough. So it began to be assumed that there was a triplet, there's a triplet code for the sequence of amino acids. In other words, three bases in the DNA coded for 1 amino acid. Now we're all familiar with those tables now of the codons in mRNA. All 64 of them. You know, for example, we know that U, that will code for the amino acid phenylalanine. UCU, that codes for the amino acid serine, and so on. We're all fairly familiar with that table. So the amount of work that went into working that out was enormous, and a lot of innovation. So we're going to look at how that was done. Okay, now the first progress was made by a scientist called Nirenberg, who was an American scientist in the early 60s. And what he did was he got he got a cell-free system for synthesizing proteins. So he got E.coli, which is a very well-known microorganism, and he lysed it. That means he broke it open. And this is called an E.coli lysate. So it's essentially E.coli cells all broken up, and everything in them is released into that medium, the buffer in there, and so on. So there'd be a mixture of ribosomes where you're going to get the protein synthesis. There'd be all the tRNAs you'd need for the translation process, and then these enzymes called amino-acyl transferases. They're the enzymes which are responsible for sticking an amino acid covalently onto the transfer RNA molecule. Okay, so he had his E. coli lysate, and he then added some RNA to that. The RNA he added was, at that stage, they could not synthesize, like nowadays you can synthesize, 30, 40 very specific base sequence of RNA in large quantities. They couldn't do that then. They could make RNA, but all he could do is he could get lots of uracil units, uracil nucleotides, and make them into a polymer, and they call that poly-U. Okay, so that would be, so if you looked at the RNA there, it'd be lots of U residues. That's all you've got, just U, loads of them. Okay, so he added that RNA, basically because that was easy to make. It was the only RNA that they could make, into this mixture here, and they were going to use that. That was going to act as the messenger RNA. So he added that. They leave this to incubate. Okay, so they leave it for 24 hours, or I'm not sure how long they left it for, in order for protein synthesis to occur. And then they added an acid called trichloroacetic acid, TCA, and that has the effect of precipitating all the protein out into a solid at the bottom of the beaker. No, it wouldn't be a beaker; it'd be a very small vessel they did it with. Oh, and then you can filter the protein there. Now, I should have said earlier, what they did, they added amino acids here. He added all 20 amino acids, and he labeled one of them with radioactive carbon 14C. Okay, so he had to do 20 different experiments here. Each time they changed the radioactive amino acid. So once with glycine, once with serine, and so on. Okay, so they did that, and then they precipitated the protein, filtered the protein, and then we're using a scintillation counter. You can count the radioactivity. So you'll give you in counts per minute these levels of 14C in there. So if there's any radioactivity in the protein, it must have come from the one radioactive amino acid that we've added there. So they did this, and they found out that when they added poly-U, and this was a real eureka moment, they found out that poly-U gave, do that up here, sorry. When they used poly-U, they got the protein that was produced only contained the enzyme, the amino acid phenylalanine. So they got polyphenylalanine there, a protein just made up of phenylalanine amino acids. And so that told them that the codon, the triplet U, must code for the enzyme phenylalanine, for the amino acid phenylalanine, sorry. So they worked out one of the 64 codons, and that was a breakthrough. So obviously after doing that, then the obvious thing to do then was to repeat the experiment, and they could only make RNAs with one with one base in it. So then they did it with poly-C. So with poly-C, they ended up getting the amino acid which incorporated there was proline. So from that, they worked out that CCC code, that's the codon for proline. They did the same with poly-G, and they worked out that that was glycine. So they know that the codon for glycine is GGG. And then the last one they did was poly-A. And that was actually a little bit difficult because, I'll tell you why in a minute, but poly-A gave you the, lysine. So AAA codes for lysine. That was a little bit difficult because polylysine, the protein, unusually, unlike most proteins, it is soluble in this trichloroacetic acid, so it didn't give a precipitate. So they didn't get any protein precipitate. So they had to precipitate out in a different way with that one. So that was one of many difficulties which they overcame. Right. So they worked out the 4 codons. Right now, let's rub this out, and we're going to now look at the next stage and what they did. Nirenberg and Matai was the other researcher. Okay, so this was all in the late 60s, the early 60s, sorry. What they did then was they couldn't make specific sequences of RNAs, but they made RNA. They got UTP, so that's triphosphate nucleotide of uracil, and they got CTP, and they mixed these together, and that made RNA molecules containing only U and C. Okay, so they've got these great big long RNA molecules, but instead of having all 4 bases in, they only had 2 bases, sorry. So if you think about that, they've randomly combined. You can get the following codons. You can get UUU. You could get UCU, CUU, and you could get all the C's. Okay, so they know what 2 of these were anyway. They looked at so they added this RNA to the E. coli lysate, and they used the radioactive amino acids again, and they found out that the only 4 amino acids which were incorporated into proteins using this RNA of U and C, they've got 4 of them. They've got phenylalanine, they got proline, and they also got serine and leucine. Okay, so what did that tell them? Well, phenylalanine, they already knew that the phenylalanine was being incorporated because you've got that triplet there. So that they didn't tell them anything new. They also, unsurprisingly, they got proline incorporated, and that didn't tell them anything because, of course, that comes from those 3 C's. They've done that from the poly-C experiment. But these two, what they could tell that they must be serine and leucine, but they couldn't tell which one was serine and which one was leucine. That was impossible to tell from that experiment. So Nirenberg then did a different experiment. Okay, he's gone as far as he could with that one, and he also did. He didn't just do that for U and C. He did it for all combinations of RNAs with just 2 bases in it. So he did get some information, but nothing definite about other codons. So he had a different approach, and what he did was, in the next set of experiments, he got a piece of nitrocellulose filter. This is a filter here. And on the filter in green there, he put some ribosomes from E. coli. And he put them on the filter, and the ribosomes are too big to pass through the filter. Then what he did, he added to the filter, he added some trinucleotides, okay? So, and he knew the base sequence of these. So they couldn't make great big long molecules of RNA at that stage, but they could make trinucleotides of any order of bases that they liked. And that took them a long time to do that, actually. And that was part of the work, was the ingenious ways in which they did that. But they were able to make trinucleotides in reasonable quantities. So he put a trinucleotide they were interested in. Let's say they were interested in UCU. They put that on, right, and that will stick to the ribosome, okay? And then also they added transfer RNAs, which were charged with a 14C labeled amino acid. Okay, so for each one of these, each thing, they'd have to do 20 different experiments, because they'd have to each time do 20 different transfer RNAs with the amino acids attached, because there's 20 different amino acids. Okay, so we put that on there. Now, the next stage here was to wash the filter. Now, if the transfer RNA binds to, if it's complementary to this, if it's got the right anticodon for that codon, it will stick to that, and that's stuck to the ribosomes, which are on the filter paper. So the transfer RNA, if they've got the right combination, would stick. Then they washed it, and that removed any unbound transfer RNA. So, and they'd have to do this with 20 different amino acids, as I said. One of them, then they count the radioactivity on the filter, and 19 out of 20 times there would be no reactivity there. But for the case of the UCU one, which actually codes for serine, you would find out that when you added 14C labeled serine attached to its transfer RNA molecule, when you added that, there was radioactivity on the filter, and that shows that it matched to the codon UCU. Okay, so they worked out, in that way, they worked out the almost all 64 of the codons. Not quite all of them. Some of these results weren't terribly conclusive at times. So there's a few that they weren't sure about. And of course, there are 3 stop codons, and no transfer RNA were bound to them, so you couldn't really identify them. So other experiments were done, and hopefully you'll watch part 2 of this video to see the following experiment, which finally wrapped up the whole genetic code.